Welcome to your pre-lab webinar on nucleophilic substitution reactions, and especially regarding the experiment we will do this coming Monday on SN1 and SN2 kinetics. We'll start by looking at the reactions and getting a general feel for how we develop them. We'll discuss the mechanisms of nucleophilic substitution. We'll discuss a little bit of the experiment that you'll be doing, and we'll talk about some tips on how to do proper kinetic assays. In general, SN1 favors protic solvents. This is because it utilizes a carbocation intermediate, which is the rate-limiting step in the reaction. Protic solvents help to stabilize the carbocation in solution. Steric hindrance tends to promote the formation of carbocations because backside attack from an SN2 mechanism is not possible due to the blockage of the sterically hindered group. It is also more usable in situations where a leaving group may not be the best leaving group possible. SN2 reactions prefer an aprotic solvent, which means that there is not a lot of dipole interaction between the solvent and carbocation, or rather facilitation of orienting different charges towards the structure. It uses a transition state intermediate, which is technically not a fully existent object, but rather a state of existence between two changing states. It is not rate limiting. The reaction is its own self-limiting endeavor, depending on the concentrations of the two compounds that are interacting in the reaction. Steric hindrance will prevent the mechanism, but if an object is not, if a substance is not sterically hindered, then the reaction will proceed much better. It prefers a good leaving group. And what we mean by a good leaving group is the relationship that objects going down the column tend to be bigger in size, and the bigger the size, the better the leaving group tends to be. Therefore, you have probably learned in class that bromine is a more favorable leaving group compared to chlorine, and that iodine is the most favorable of the halides that are used in terms of leaving groups. However, iodine also tends to move backward as well as it moves forward, which is why it is not the most commonly used leaving group. In order to review the mechanism, we must first understand the physics behind some of the chemistry we're doing, which has to do with the polarity or the dipole interaction between the carbon and the halide group. The halide is more electronegative than the carbon, and this tends to create a dipole moment between the carbon and the halide. We always draw our arrows towards the more electronegative structure in chemistry. In physics, you have no doubt learned that in the same sort of way, an electrical field tends to establish in the direction from positive to negative, or the area of highest potential to lowest potential along the gradient. It is the same principle here, except that we tend to use electrons as our guide as to what is the lower potential in that kind of relationship. This dipole moment that's created is subject to being attacked by nucleophiles, which have a lone pair that they present as being able to interact with this species. In some cases, this will undergo heterolytic cleavage, yielding the halide ion. Again, this depends on the specific mechanism. This can be somewhat of a generalization when we say it such as this. Here we see a review of the mechanism. The first mechanism shown is for the SN1 reaction. This is where the alkyl halide is placed in presence of protic solvents and acids in most cases, such as dehydrating acids or in some cases some type of solvent that will allow for a proton to be donated to it to help the carbocation form. And in that process, there is a rate limiting step, and so the rate tends to be governed by formation of the carbocation. A nucleophile can then interact with the carbocation and form a new bond between the two. This forms a racemic mixture. In the second example, a nucleophile is presented with an alkyl halide, and a transition state occurs in which a backside attack by the nucleophile creates a new product with a leaving group coming off on the forward direction. Because of the nature of this mechanism, the SN2 reaction, as this type of mechanism is so termed, is stereospecific. This does not necessarily mean that the object made, or rather the substance made, will have stereospecificity at all times, since not all reactions produce chiral centers. However, in many cases, it is true that there is a chiral center, and chiral centers are affected by these types of mechanisms. And you must be able to keep track of this if presented with a problem where stereochemistry becomes important. It is important to remember that there's no intermediate in this process. It's just a transition state. And so it is the reaction itself that creates its rate limit. In the lab exercise you will do on Monday, we're going to examine these different mechanisms and see what the structural effects on alkyl halides do to these different mechanisms. We'll do that by using solvents that promote those two mechanisms on a set of chosen alkyl halides. And we do this for each one in order to see if reactions will take place. Some reactions will take place faster than others, some will take place at similar speeds, and some will seem to never happen at all. It depends on the structure of the alkyl halide and the type of system that we use for that particular reaction that we set up. Questions you'll want to think about will be why these choices of reagents favor the different types of reaction processes that are going on. We can discuss these more Monday, but in the meantime, some basic thoughts on this would be to go back about 2 slides from previous and review the general guides to nucleophilic substitution reactions and try to understand what those are really saying to you. For instance, here we have ethanol and silver nitrate. Another good question to think about would be, why might we use silver nitrate, and what role does silver have to play in this sort of mechanism? And an example of this, and to kind of answer some of this question, would be that ethanol is a protic solvent because it has hydrogen bonding and a nice dipole to it. So it's going to really help to stabilize the carbocation. In the case of silver nitrate, we know nitrates are typically pretty water soluble. Not that we tend to place water in this type of reaction. These are organic solvents, but we also know that of the two items that would be present, especially since ethanol has some water in it, and so does silver nitrate when it's in solution, we would expect to see that silver would precipitate, which is what this reaction is indicating. So remember that silver halides tend not to be very soluble and tend to fall out of solution. And an advanced general chemistry concept that this brings back into play is Le Chatelier's principle, which governs equilibria in chemical reactions and becomes important when doing kinetics assays. So these are all things that you should think about when looking at how these reactions work. And you should especially take note when something is being done to drive something forward. Down here, you see the setup for the second reaction, which is the sodium iodide and the acetone. Acetone is not quite as protic as ethanol. You can't remove a proton from acetone very easily. And even though it has some polarity to it, it's not quite as polar as the ethanol molecule is. Also, sodium iodide is an excellent leaving group, which can be used to, well, it's both a good nucleophile and a fairly decent leaving group. But in any case, what we're actually doing here is making a new alkyl halide. You can see by placing the iodine in place of it. And we're driving the others out because those will be less soluble with the sodium in the acetone. So these are things you want to think about, is the solubility of the sodium iodide and acetone versus the sodium chloride or the sodium bromide that might form from different alkyl halides. So always think of questions like that. Think when you're working with these reactions and you're thinking about mechanisms, really try to get into how you set them up. And we're going to talk more about how SN1 and SN2 are favored versus E1 and E2, because it tends to be the case that SN1, SN2, E1, and E2 have this connection with one another. And in any sort of case, depending on how you tweak solvent parameters and what kind of structure you're working with, one of the four or two of the four, sometimes in certain cases, may be more favorable than one alone, or all of them alone, or I mean all of them together. So you have to look at it from those angles, and we'll go over when the time comes, when we get closer to working with E1 and E2, how all of this ties in with one another. For now, we just want to focus on SN1 and SN2 and try to get that much understood about the differences in solvent systems. Further information on the experiment we'll do shows some of these reactions again that you'll be using here, just to reiterate so you can see what you'll be working with, making this soluble agent here. Notice these don't actually bind. We're making a sort of ionic solution in this particular case, because that's going to help stabilize this. And here we're just doing an exchange. Screw cap tubes will be available. There are these small centrifuge-like tubes, scintillation tubes, sometimes they call them. Sometimes they remind me of culture tubes from the microbiology lab, but those tend to be just a little bit bigger. But anyway, you want to make sure that you clean and return these when you're finished using all of these things. We don't want any dirt or residual chemicals for the next group, so you do have to clean these tubes when you're done and you've disposed of your waste. The alkyl halides will be in the barrel pipettes, which I'm not sure we've used these yet with you all. I can't remember if we have or not. We've done so many different things. I don't think we used the barrel pipettes yet, but they're basically large plastic pipettes. And our general rule of thumb is that about 4 drops is equal to 2/10 of a milliliter. So that would mean that about 10 drops or so is somewhat equivalent to a mil of volume. Is that really accurate? No, it's not super accurate, but we don't have to be spot on the nose for this. We just have to have a good rough estimate of about how much volume you're putting, because we just want you to kind of get rough numbers for this and see the difference. We're not looking for a full-on quantitative kinetic kind of output from these particular set of experiments that we're doing. Let's see. As far as, knowing whether a reaction went forward, because of the way we've set it up, we want you to recognize that you'll probably see some precipitate form in the tube, since the things we're putting together are making these not as soluble when they come out, and so they tend to drop out of solution, and that removes them from the equilibrium contribution, which means the reaction will drive forward. And again, make sure you know which solvents favor which substitution system. Some other tips I'll give you about kinetics assays are the following. You need to make sure that you're being very regimental and very methodical in the way that you set up these experiments. You want to be cautiously quick with getting things into the tube, closing them off, and beginning measurements of the time that's elapsing when you start dropping reactants together. And this is because you don't want any lag between the time you start recording and the time that you begin reacting. So it can be good sometimes for you to work with another person if there's not enough room in the specific area, or if you're having to work in the water bath, and to work together to make sure you're recording your time, since everybody can't always have their cell phone at the same time right in the same place. Most people use their cell phones and the stopwatch functions that are on them now. You don't have to. You can use a regular watch and simply record the time. However method is best for you. Just remember that you're looking at the elapsed time the reaction occurs, not, necessarily like this hour versus that hour. It's the difference between the start and end time, the total time that you need. You also, going in, kind of want to think about the reactants that are involved and kind of have an idea of what your rate law might sort of look like, so that when you get these numbers and you start determining your rate laws, you'll know what to expect out of the data set, and you'll be able to interpret the data better. And remember that we generally base those rate laws off of the elapsed times and the concentrations of the substances involved in the reactions. And that's pretty much all there is to this one experiment. It's a pretty short experiment. It doesn't take too horribly long to do, and we're looking for a very simple thing, just the differences in time and how we can compare these different reaction systems to see why certain things are said to be more favorable than others. And you can actually see this for yourself. So it's a good start to reactions. This is more of a kinetics assay, but the next experiment we do will start getting into reactions and different kinds of products we can expect to form. So it will get more complex as we move through them, but this is a good beginning, and everyone should not stress too much over it. So we'll see you Monday, and have a good weekend.